So hello all, I'm starting with the second lecture of Principles of Inheritance and Variation. So in the previous class, we talked about the concepts of monohybrid cross, right? Like what is monohybrid cross, how to do monohybrid cross using a Punnett square, right? Then we also saw two laws of inheritance based on this monohybrid cross. These were given by Mendel. Right. Now, in this class, we will continue our discussion on Mendelian inheritance, like on using the concept of dihybrid cross, and also we'll look into what are the exceptions of monohybrid cross and what are chromosomes. So these all concepts will be covered in today's lecture. Okay. So Mendel gave two laws is based on monohybrid cross. One is the law of dominance, another is the law of segregation. So if you recall, what is law of dominance? If the organism is of heterozygous condition, that is containing two dissimilar alleles, so one allele will be dominant over the other, such that only the dominant allele will be expressed and the phenotype corresponding to that dominant allele will be seen. Okay. Whereas law of segregation talks about how the alleles, the two alleles, get separated during gamete formation, right? So if the heterozygote condition is there, one type of allele will be present in 50% of the gamete, another type will be present in another 50%. And if it is homozygous, then all the gametes will have the same allele, right? So these are the two laws. Now, there are two exceptions which are seen in nature, which doesn't follow this Mendel's law. First is the incomplete dominance. So what is incomplete dominance? So this phenomena was observed in inheritance of flower color in a plant called snapdragon plant. Okay. So here it is. So this is how the flower look like. So what this saw is the dominant allele is not completely dominant over the recessive allele. So if you see this monohybrid cross, right? This is the red color flower, okay? This is the white color flower. Both are homozygous. They are pure line. They are coming from the pure line plants, okay? So they produce only one type of gametes. So in the F1 generation, although it is heterozygous and capital R here was dominant. Okay, that's why it is written in capital letter over the small r. But we don't see red. We see pink color flower. So the F1 progeny doesn't resemble either of the parents. It is showing a phenotype which is in between. Okay. Similarly, when they did selfing, okay, self-pollination, in F2 generation, they saw the plants which were heterozygous, they were all pink, whereas the homozygous plants, they showed the same phenotype as their parents, right, at the parent generation. And the typical phenotypic ratio of 3:1, which was observed during monohybrid cross, was not the case, right? Instead, we see 1:2:1. This is the phenotypic ratio. Here we can easily distinguish the heterozygous plants from the homozygous ones because these heterozygous plants are having a pink color flower. So when this is the case, uh, this type of phenomena is called incomplete dominance. So as it is written here, the F1 generation plants, they had pink flowers, okay? And these flowers did not resemble either of the two parents, but it was in between the two. So I think incomplete dominance, the concept of incomplete dominance must be clear to you. The F1 progeny did not resemble any of the parents. Rather, it showed a phenotype which is in between. Okay, the dominant allele is not completely dominant over the recessive allele. Now, let me, in more elaborate manner, let me explain what are genes and what are alleles. Okay, we have already discussed the definitions in the previous lecture. So you already know that genes, they contain information for expression of a particular character, right? Like flower color. So in this case, there's a gene that is responsible for giving a color to the flower, and that color can be either red or white. Okay, so what type of allele is present will decide whether it is red color or white color. So what is allele then? Allele is a slightly different version of a particular gene. Now in diploid organisms, two copies of gene exist. Okay, so one gene, they will have two types of alleles. So pair of allele will be there in diploid organisms. But in heterozygous, the alleles, they are not identical. In homozygous, they are identical. These things we already know from our previous lecture. Now consider this example. Okay, so suppose there is a gene A and it codes for any enzyme. Okay, and this enzyme is participating in any biochemical reaction. So you know that enzyme, when it reacts with a substrate, it gives some product. Okay, so this enzyme must be participating in a reaction, biochemical reaction, where there is some substrate associated with this enzyme. Now this gene is known to have two alleles. Say the allele, we can name it as allele A1 and allele A2. So consider this case, the normal case. Now if A1 allele is expressed, it will give the enzyme A1 with a structure something like this. And this is the structure of the substrate. Then it can easily fit with the binding site. This is the binding site. So ultimately it will give a product. So product is formed from the expression of A1 allele. Now another case, consider an abnormal case, okay, where due to the expression of allele A2, another enzyme is expressed, enzyme A2, but the binding site is now different. So this binding site is not compatible for binding the same substrate. So because of which product is not formed. So you can see that one allele is giving an expression of one type of enzyme, whereas another allele is giving an enzyme which is not at all functional, which is not at all doing its job, right? And if it is heterozygous, if both the alleles are present, and means in a heterozygous individual, it is like we say that, or it is seen that allele A1 is dominant over allele A2. All right. So if it is heterozygous, like I said, then the normal phenotype means we will get an enzyme which is producing the desired product when it is reacting with this corresponding substrate. But when it is homozygous for allele A1, then it's also normal phenotype. Problem comes, or when diseases happen, when allele A2 is homozygous. Okay, so allele A2 is recessive in nature, and when both the copies of allele A2 are present, we get an enzyme which is not at all functional. So if a non-functional enzyme is expressed in an individual, the biochemical reaction will stop there, and whatever desired product which is happening in normal case is not going to like present in that individual. So some particular abnormal phenotype will result, right? So now you can understand like the combination of alleles, like how much it matters, and like how it can give rise to any particular disease. This is one just simple instance of how it can happen. So how alleles are actually formed, okay, from a particular gene. So this it will be discussed in the next chapter, okay. But now, for now, I can tell you that within the allele there is some changes, okay. There is some changes in the gene which creates the particular allele. What type of change, where that change happens, this will be discussed in the next chapter. Now there is another exception, and this exception is called codominance. Okay, so what happens here? Both the parental alleles, they are expressed together in F1 offspring. Okay, because of which they resemble both their parents. Means allele that is coming from both mother and father, they are expressed together in the offspring, because of which the phenotype of that offspring resembles both their parents, both their mother and father. A very good example of the codominance phenomena is ABO blood groups in humans. Okay, so you're all aware that we have particular type of blood groups, right? Like I am B positive. Similarly, someone will be A or someone will be O. So how that actually happens? So it has a genetic explanation. Okay, how an individual gets a particular type of blood group. Now, it is known that ABO blood groups, they are controlled by a gene called I. Okay, and this gene I, it contains the information for the expression of a sugar polymer. And this sugar polymer is present on the cell membrane of the RBCs or the red blood cells. So whatever type of sugar polymer is present on the RBC, that will be the type of the blood group. Okay. Now this gene I, it has three alleles: IA, IB, and small I. Okay. So you can see this is an example of multiple alleles. Like one gene doesn't necessarily have to have only two alleles. It can have three or four, like various kind of alleles for that particular gene. Okay. Now, people saw that when IA allele is expressed, then type A sugar is expressed on the RBC. Similarly, when type B allele is expressed, type B sugar is expressed on the RBC. And when small I is expressed, then there is no expression of sugar on the RBC. That is, there is no sugar polymer found on the surface of RBC for small I expression. Now humans are diploid, we know that already. Okay. And so any individual will have only two of these three alleles, right? Only two of these three alleles. And it will decide what will be his or her blood group. So in how many ways can you combine these three alleles? Okay. So that you get different types of phenotype. We'll now check that. Now there can be homozygous cases where two copies of IA is present. So it will result in type A blood group, right? Both copies of IB allele is present. Then type B blood group will be present. Okay. And if both copies of small I is present, then the blood group will be O. So three genotypes till now, all are homozygous in nature, and there are three phenotypes, A, B, and O. Now, there can be heterozygous cases as well. So, for understanding heterozygous case, we have to first know, in order to say what will be the phenotype for a particular heterozygous case, we have to first know which gene, which allele is dominant over which allele. Okay. So, if the heterozygous case is IA, small I, then it is known that IA is dominant over I. So, the phenotype will be A. If you have guessed it, you are correct. Okay. The phenotype will be A. Similarly, the heterozygous case where IB and small I together are present. So in that case, IB is dominant over small I. Okay. So if it is dominant, then the individual will have B blood group. But what if both IA and IB are present? Now it is seen that these two alleles are both dominant over each other. Okay. Neither are recessive to one another, both are dominant. So this is a classic example of codominance. So the individual will display both type of sugars A and B on the RBC. That is why the person will have ABA blood group. So from this workout, we can see that there are six types of genotypes. Okay, there are six genotypes, and the phenotypes, that is what type of blood group that individual have. There is 1, 2, 3, and 4. So there are four phenotypes or four types of blood groups an individual can have. Okay, any of the four. Okay, not all the four together, any of the four, because an individual can have only two copies of these among these three alleles. Now, how did people find out, like so there are three alleles? How did they do that? So actually people do, there's a field called population genetics, where people study a group of individual within a population. Okay, so like India's population, people are studying. So from while studying, they saw that for this particular gene, that gene that is corresponding for the blood group, it has 3 alleles. Okay, so this is an example of multiple alleles. Similarly, there can be like 4, 5, 6 alleles for any type of gene. Okay, you can get examples like those as well. Another like example or an exception to Mendel's inheritance, like Mendel's laws of inheritance, is that a single gene product, it can give rise to more than one effect. Okay, effect means phenotype. It can give rise to more than one phenotype. Okay, which was not the case with Mendel's monohybrid cross, right? If there is a allele for round seed shape, only the round seed shape will be present. But it is possible for few exceptional genes that it can give rise to more than one phenotype. So this is an example for that. Okay, suppose in a pea plant, means in a pea plant, people have saw that there are 2 alleles for a gene, capital B and small b, the gene that controls starch synthesis. So for starch synthesis, there is a gene and it has 2 alleles, capital B and small b. And capital B is dominant over small b. So when it is homozygous for capital B, okay, then the starch synthesis will be there, so it will produce small grains. So these grains, they are large, okay, and also it gives rise to round shaped seeds. The seeds are round, okay, for this capital B, that is homozygous condition. Whereas for homozygous condition of the small b allele, okay, the starch grains, they are small, okay, and the seed shape, it is also affecting the seed shape, it is wrinkled, okay. But when in the heterozygous state, we see that the starch grains, they are of intermediate size. It is also a phenotype, the size of the starch grain. So it is this the size of the starch grain it is not resembling either of the parent type, right? Either of the capital B, that is homozygous type. It is not resembling either. It is of in between size. So this is a incomplete dominance is happening. Okay, incomplete dominance is happening for this particular phenotype. But when we see the size of the like shape of the seed, it is round for the heterozygous one. So in this case, dominance, whatever concept of dominance, that concept is applicable here. So for 2 different phenotypes, that is resulting from the expression of only one gene, okay, and we see that in this example, two type of like phenomena is happening. In one phenotype, incomplete dominance is seen for the heterozygous condition or the hybrid plant. Whereas when we consider the shape of the seed, that is whether it is round or wrinkled, it is showing the dominance, right? The capital B is dominant over small b. That thing is applicable. Okay, so these were 3 phenomena which are exception to the Mendel's laws of inheritance, which he gave for the monohybrid cross. Now let's quickly move to doing a dihybrid cross. So what is a dihybrid cross actually? So dihybrid cross deals with the inheritance of 2 genes. Okay, and okay, 2 genes. What happens? Mendel has taken 2 pea plants. So Mendel has taken 2 pea plants, and these 2 pea plants, they were they were same, completely same, except for 2 characters. They differed in only 2 characters. So consider a pure line pea plant with round and yellow seeds. Okay, and another pea plant, it is also pure line, which is having wrinkled and green seeds. So here 2 characters are chosen. One is the shape of the seed. It can be either round or wrinkled, or another is the color of the seed. It can be either yellow or green. And these 2 pea plants, these 2 pure line pea plants, which will be using as the parental generation, these 2 pea plants differed in only these 2 characters. Other characters were exactly same. So when we are setting up this kind of an experiment, then it will be called a dihybrid cross. Okay. Now let's annotate like how are we going to represent the alleles. So for the gene or for the character seed shape, two alleles will be used. One is capital R, which represents the round shape of the seed. Another is the small r, which represents the wrinkle shape of the seed. Now, another character, seed color, will be using capital Y to represent the yellow color of the seed and small y for representing the green color of the seed. Now, from monohybrid cross, Mendel saw that round round shape is dominant over the wrinkled shape, okay? And yellow color is dominant over the green color. So this type of a thing he saw from monohybrid cross when he was considering one character at a time. Now see, now let's see whether he got that from dihybrid cross as well. So this is the parental generation, right? Suppose we choose a male plant, which is a pure line, right? And another, so female plant is pure line. And this is pure line for the round shape and the yellow color. And another pea plant, which is homozygous for the wrinkle shape of the seed and green color. So he crossed. Okay. So each of these plants will create gametes, like male one will create pollen grains, female plant will create eggs, right? And the creation of the gametes, whatever the alleles will be present, uh, we will use law of segregation to write the genetic combination of each gamete, right? As you know, uh, when the alleles separate during meiosis, so one copy of the allele goes into each gamete, right? So RY, this is the gamete from the male plant, this is the gamete from the female plant. So in the F1 generation, this is the F1 generation, what we see is the genotype is this: capital R, small r, capital Y, small y. This is the genotype of all the F1 plants. So what is the phenotype? He saw that the phenotype was round and yellow seeds. So from this he concluded that whatever he saw in the monohybrid cross, that round shape was dominant over wrinkle shape and the yellow color is dominant over green color, is also true for dihybrid cross, right? Round is dominant over wrinkled, yellow is dominant over green. This is what he saw from the F1 generation. Now for the F2 generation, you know what to do. You have to do selfing between two plants from the F1 generation. So these are the two plants. Suppose one plant is male, another plant is female. And first you have to write what is the genetic combination of each gamete. So here I have written 4 types of gametes each plant is producing. But how to understand what will be the genetic combination of each gamete? How have I written? So for writing this type of genetic combination of gametes, you have to use Mendel's law of independent assortment. So this is a new law which I'm explaining just now. Before, in the monohybrid cross, this law was not like proposed. This law is only comes when you are doing dihybrid cross. So according to this law, in hybrids or in heterozygous organisms where they have two different pairs of alleles, okay, it is for two genes. So this is a hybrid, right? Which is having heterozygosity for two genes, okay, for the seed shape and seed color. So in these type of plants, segregation of one pair of allele is completely independent of the pair of alleles of the other pair of alleles or the other gene. And the segregation happens during meiosis. Okay, so how capital R small r is segregating is completely independent of how capital Y small y will segregate. Okay, these are totally independent events altogether. That is what this law says. And using this law only, you can understand how the gametes are formed. Okay, so this is the parent, right, which is heterozygous or hybrid parent. So it will have it will create gametes. So let's consider for this gene. Okay, for this gene, one copy of allele capital R will be passed on to 50% of the gametes, whereas the small r, this allele will be passed on to another 50%. Okay, so how this capital R and small r, they are passed on to each gamete is not affected by these with this pair, right? Capital Y, small y. Now these two uh, types of gametes should also have alleles for the color as well. Okay, so one gamete will have one copy of every type of gene, one copy of the allele for every type of gene, right? So this type of gamete, which is having capital R, now it will have 50% chance of getting either capital Y allele or small y allele. Same is true for the gamete which contains small r. It will have 50% chance of containing capital Y allele, as well as like another 50% chance will be for small y allele. So if you do simple analysis like from this diagram, you can see that capital R, capital R, capital Y. This is one type of gamete that is formed. Then capital R, capital small y. This is another type of gamete that is formed. Small r, capital Y. This is another gamete. Small r, small y. This is another gamete. You can simply do like this. Capital R combined with small capital Y. This is one type of gamete. This is combined with small y. This is another type of gamete. Now you are done combining for capital R. Now do the same for small r. Small r is then combined with capital Y. This is another type of gamete. And small r is combined with small y. This is another type of gamete. So same thing, same result is obtained from two types of explanations. So after doing this only, I saw that four types of gametes are possible from each parent. The genotype of each parent is same, right? So same types of gametes are obtained from both the parents. Okay, so if there are 100 gametes, 25% of the gamete will be capital R, capital Y. Then like this, we can say that there is a chance of 25% of arising of a gamete having capital R, capital Y. Okay, same goes for each type of gamete. So we will use these gametes and like put it in a Punnett square like this. So this top row is for the gametes coming from the father. Left column is for the gametes coming from the mother. Now, if you have a pen and paper, okay, you can do it yourself as well while I'm doing it. You just have to combine the different genotypes. Capital R, capital R, capital Y, small y, capital R, small r, capital Y, small y, okay, capital R, small r, capital Y, small r, capital Y, capital R, small r, capital Y, small r, capital Y, small y. So you have to carefully look at the rows and columns while writing. You can easily make mistakes if you don't focus while writing, okay? It is a bit complex than the monohybrid cross because there are 16 squares. So these are all the 16 types of genotypes which are possible. So we can then quickly write what are the phenotypes possible. Both copies of capital R are present. The seed will be round, and both copies of capital Y is present, so it will be yellow, round and yellow. Similarly, we know capital R is dominant over small r. The round shape is dominant over the wrinkle shape. So when it is heterozygous for this gene seed shape, like in this case, the phenotype will be like this: round, yellow. Here we know that yellow color is dominant over green color. Okay, so when it heterozygous for the gene seed color, we know it will be a yellow color, round yellow, round yellow. Now homozygous, now there is homozygosity for the recessive gene. So seed shape will be wrinkled, but the color will be yellow, round yellow. Again, it will be wrinkled and yellow, round yellow, round yellow, round, but it will be green because it is homozygous for the allele corresponding for green color of the seed. Round, then green color, round, yellow. Here it will be wrinkled, right? Wrinkled and yellow. I hope you can understand why it is wrinkled yellow. And here will be round and green, then wrinkled and green. Okay, so these are the different types of phenotypes. Okay, so if we were to see at this Punnett square and determine the phenotypic ratio, so there are four types of phenotypes if you carefully observe. Round yellow, correct? Round yellow. Then there is wrinkled yellow, correct? Then what else? Is round green and wrinkled green. Now see, these two are the parental types, and these two type phenotypes are new, right? These are not present in the parent. So if we were to count, then we'll count the number of boxes where these phenotypes are present, then we can get the ratios. Okay. So for round yellow, how many boxes are there? 1, 2, 3, 4, 5, 6, okay, 7, 8, 9. Okay, so it will be 9 is to now wrinkled yellow. Okay, 1, 2, 3. 3 is to round green. How many boxes? 1, 2, 3. And for wrinkled green, there is only 1. So this is the phenotypic ratio that you have obtained from this Punnett square. And this is also the same ratio which Mendel obtained. But what is the genotypic ratio then? Can you determine? So as you can see from this Punnett square, it will be not like, a small ratio. It will be quite big. We can see so many types of genotypes, right? Uh, so let's write all the types of genotypes, and from that we can get genotypic ratio. The first type is this. How many boxes are there? One, right? One, and okay, only one. Okay. Then this is one genotype, right? Second box. This is another genotype. So like this, you can count the boxes corresponding to each type of genotype, and then write one by one and get a ratio like this. So this is your task to do it. It's quite easy if you have understood like how I have derived at this Punnett square. Okay, how the phenotypes are assigned. Okay, and after that your job is done. You just have to observe carefully and write it down, and count how many boxes are there in the Punnett square, and you will get the ratio automatically, just like what we did for the phenotypic ratio. So this is all about dihybrid cross. Now moving on, let's familiarize with ourselves a little bit of history. So what happened after 7 years of work, Mendel published his work in 1865. Okay, and it was not recognized, like the importance of his work was not recognized until it was 1900s. Why? Because his work was not easily available. There was no internet, right? We could not search for it. Also, his work was not widely publicized among the like general public. People didn't knew about it. Only few experts in the field knew about it. And these experts were not very happy with whatever concepts he gave. He told, right, the genes, or the in his term, in his words, the factors. These are stable discrete units, and these units do not explain how the variation is observed in the nature, as he showed that these genes, they do not blend with each other. So if there is no blending, how they are actually resulting in the variations, right? The parents and the offsprings, they do not look exactly the same, as well as the different siblings, they look different from one another. So this type of variation, from where it is arising, his work didn't explain that. Also, it was not acceptable at that time for biologists to use maths or mathematical techniques in biology. They didn't like it at that time. But in 1900s, okay, people started discovering more and more about the cell and the nucleus and what are chromosomes. Okay, there were like high power microscopes available at that time, through which we could see the structures, that are chromosome structures, we could see in the nucleus. Okay, in 1900s, by then it was developed. So while looking at the chromosomes, people have understood the concept of meiosis, mitosis, right, that we have studied the chapter cell cycle. Okay, these are different type of cell divisions that happen. So people have understood. So at that time, in the 1900s, at that time, his work was again rediscovered by few scientists. Okay, and they tried to repeat his work and give credibility to his work, and applied whatever results he got in their own work. So these two scientists, Sutton and Bovary, these two scientists actually saw that they knew about what are chromosomes. Okay, so they saw that how the chromosomes are behaving. There's a similarity with the behavior of the genes. Okay, which Mendel proposed, right? This behavior of the genes, like the alleles, the pair of alleles exist for each genes, and how they segregate. So, and his laws, these type of behavior that Mendel proposed, it was similar to what he has observed, they have observed for the chromosomes. Now, if you recall the concept of meiosis, which shows like uh, during each step how chromosomes are segregating, okay, then pairing, okay, and forming the haploid sheets. If you recall those concepts, you can understand why are they saying that. Why are they saying that the behavior of chromosome is similar to the behavior of the genes? So here is one cell, okay, and at the beginning of the meiosis one, if you recall, in prophase one, the cell, the DNA content of the cell, it has doubled, right? And now we can see the condensed form of DNA, that is the chromosome. These are two chromosomes, and these two chromosomes, they are homologous to one another. Okay, so they are not exactly identical, but they are similar, and each homologous chromosome come from one parent. Okay, so this cell, within this cell, say this red chromosome has come from father. Okay, or paternal. Another chromosome, it has come from mother. Now when we zoom in and see what happens to this homologous chromosome in subsequent steps of meiosis 1, if you recall, um, these two homologous chromosomes, they pair, right, and form what is called a synapsis, which is also called a tetrad, right, if you recall. So these all things we have studied already in class 11, which I'm just revising, okay, for your convenience. So here, this structure is now called tetrad because it has four sister chromatids: one, two, three, four. This is one sister chromatid, and this is another sister chromatid. So sister chromatids, they are joined together at the center using centromere. Okay. Now there's a step called crossing over, right, or recombination, where exchange of some chromosome material or the DNA content happens. So this segment of the chromosome has come from this yellow color homologous chromosome and vice versa. So at the end of meiosis 1, you get two cells, okay, where the homologous chromosomes, the pair of homologous chromosomes, it has separated. And these two cells now enters meiosis 2, where at the centromere, the sister chromatids now separate and gets distributed randomly. This is a random process. Gets distributed into 4 daughter cells. Okay, so this is the reduction type of division, which are already familiar, right? From 2n chromosome number n, all the daughter cells, they have n. So this is how the chromosome moves, right? Like how it pairs and how it segregates. So they saw that uh, similar kind of things also happens for gene, which is proposed by Mendel. So genes, they occur in pairs, right? For a gene, there is a pair of alleles. Similarly, for chromosome, right, a homologous chromosome exists, that is two similar chromosome, one chromosome coming from the mother, another chromosome coming from the father. So this property is similar for both genes and chromosomes. Another is Mendel said that genes, they segregate, right, during gamete formation. Right. Alleles, in the pair of alleles, their alleles segregate. That is under the law of segregation. He gave that law, which is what also happens for the chromosomes as well, right? You can see that the homologous chromosomes, they have segregated, and then they have later all segregated to, form haploid cells. So this segregation also happens for the chromosome. So this property is also similar. Another thing is segregation of one gene or a pair of homologous chromosome is independent of other pairs, right? So how one gene is segregating among the gametes uh, is independent of the other gene, right? Which we saw for a dihybrid cross. And the same is true if suppose there was another homologous chromosome, another pair of homologous chromosome, how they are segregating, okay, then how they are forming the synapses and all. That is also an independent event, like they are doing it independently of the other. So these three properties, right, they are similar for both genes and chromosomes. So because of these similarities, Sutton and Bovary, they then proposed that is called chromosomal theory of inheritance. Okay. Now, this chromosomal theory of inheritance, it actually united the knowledge of chromosomes behavior. Okay. So how chromosomes are segregating and pairing with the knowledge of Mendel. So they have combined these two concepts into one umbrella of theory that is called chromosomal theory of inheritance. Now, using this theory only, they argued that the pair of alleles also lies, the pair of alleles, they actually lies on each homologous chromosome, because of which whenever the chromosomes are segregating during meiosis or mitosis, the alleles are also getting separated, right? So thus when homologous chromosomes separate in meiosis, alleles also segregate. So that is their argument, and we have later on scientists, they have worked out and proved that this is indeed true, okay, using experimental studies. So till now we have covered what is dihybrid cross, then who all proposed chromosomal theory of inheritance, okay, what it is based on, right? It is based on how genes, the behavior of genes and chromosomes, they are similar. Okay, and also we saw few exceptions that are seen in nature, that are exceptions to the monohybrid cross which Mendel gave. Now, we are going to do an example problem which is based on dihybrid cross. Okay, I urge you to also do it along with me. So, for in this example problem, let's read. You are given or you are breeding two organisms, okay, and they show differences in expressing two genes. One gene is for hair color and another gene is for eye color. Okay, The father, okay, so we are going to breed two organisms. One is supposed to be father, another is mother. So next we are going to understand what are the characteristics for the father. The father has brown hair, okay, and blue eyes, whereas mother has black eyes and brown hair. Father is homozygous for brown hair allele and heterozygous for eye color gene, whereas mother is homozygous for black eye allele and heterozygous for hair color gene. Okay, now assume that brown hair color is dominant over blonde hair color. So there are two types of hair color, brown and blonde. Brown hair color is dominant over the blonde hair color, and blue eye color is dominant over black eye color. So we have to perform dihybrid cross, okay, between these two organisms and list all the possible phenotypes and genotypes that we obtain in the F2 generation. Okay, now let's just list what are the alleles first. So for hair color, there are two alleles, correct? Brown, that is represented with capital A, allele that is when it is expressed give brown hair color, and another is blonde, represented with small a. And for eye color, there is blue eyes, represented with capital B, and there is black eyes, small b. So here we are asked to assume that brown hair color is dominant over blonde. That's why I'm using capital letter for representing the dominant allele and small letter for representing the recessive allele. Now, what are the genotypes of the parents? Father is homozygous for brown hair allele. Okay, homozygous, and heterozygous for eye color allele. This is the eye color heterozygous. Capital E. This is heterozygous. Okay, this is representing the hair color. This is representing the eye color. Now it is crossed. This is the father. Now what about mother? Mother is homozygous for black eye allele. Black eye, and heterozygous for hair color gene. Heterozygous for hair color gene. So hair color gene, it is heterozygous. Homozygous for black eye color. Okay. For father, he is homozygous for brown hair allele. Okay, and heterozygous for eye color. So these are the genotypes of the parental generation. Now, what will be the gametes, right? So one gamete will be of this type. Another gamete will be of this type. Agreed? Agreed. Now here, one gamete will be of this type. And another gamete will be of this type. Let me write it like this. Okay. Then we'll do F1. What will be F1? So for that, we have to draw Punnett square. Top row, I'll write the gametes for the father. Here are the gametes. Here, left column, writing the gametes for the mother. Okay. And then do what we have been doing all along. Do combinations. Okay. So here, brown hair and blue eyes. Okay. Then few progenies will have brown hair and black eyes. Okay. Then heterozygous, brown hair and blue eyes. And here few progenies will have brown hair and black eyes. So 50% is having, brown hair, blue eyes. And rest 50% is having brown hair, black eyes. Okay. Now, this is the F1 generation, ratio for the F1 generation. Now we can choose any of the uh, offspring, okay, any of the two offspring having the same type of genotype, okay, to do F2 generation. So it will be a long calculation actually. So one case, say case 1, we are choosing two offspring with genotypes. This another case, two offspring with this genotype. Case 3 is this, and case 4 correct. So you again have to determine what are the different type of gametes possible for each cases, okay, and then draw Punnett squares to determine what are the possible genotypes or phenotypes of the offsprings in F2 generation. So I'm doing for only for case 1, and for rest 3 cases I'm asking you to do it yourself, okay. So for case 1, what are the gametes possible? This is one gamete, another is this. Agreed? Here also same. Then again, you have to draw Punnett square, okay. But what I see is for the F2 generation, the gametes produced are exactly the same, right? Like it was produced for the parent. So for F2 generation, you will get again the same result, okay. So similarly for case 2, case 3 and case 4, you have to do it yourself. And for each case, you have to determine what are the possible phenotypes. So here it was 1:1 for the phenotype, and for the genotype, it was all the genotypes, they are different from one another, right? So 1:1:1. So it will be like that. Similarly, for F2 generation, you have to do the same, determine the ratio for the phenotype and the ratio for the genotype. So that's it for today's class. I hope you have understood the concepts. I'll see you in the next one. Thank you.